My name is Ivaila Georgieva, and I'm a PhD student here at the Liverpool CMR in Professor Keogh's group. My research is focusing into discovery metabolomics and in different conditions, and the first one to look at will be preeclampsia. The main purpose is to look for biomarkers in clinical serum samples. Serum samples are very complex. The biggest challenge is to have a good picture of everything from high abundance to low abundance compounds, because we don't know which one will be interesting. It could be something that it's very easy to pick or very high abundance, or it could be something that it's relatively of a small abundance and well hidden behind something else. I'm lucky to work with a thermoscientific Orbitrap IDX, which allows us to take a great perspective from the diversity of the compounds. So personally, I'm quite new in mass spectrometry. However, I have looked at different techniques and I have worked with other instruments, and Orbitraps are indeed providing an excellent accuracy with a sub-ppm precision that allows us to be very precise into identifying the empirical formula of compounds, and especially unknown compounds. The automation of AcquireX and going into different level of a very clean DDA spectrum of different abundance and creating this exclusion list takes time, but a time that no human is involved in. So we are very happy. Using MSN capabilities, we have the opportunity to understand the fragmentation profile of substructures also of the molecule and have a better appreciation through similarities of what the structure of this molecule could be. When we're running clinical samples, and especially if we have the chance to work on a large study, we will have a lot of samples. The speed and the run time of a method becomes very important. So one of the things that we are doing here right now is to try to reduce the run time of our method from 15 to 10 minutes. This will allow us to increase the throughput of the instrument. And the reason why we can do this is that Orbitraps provide us this good resolution of multiple compounds. For our processing workflow, we use the thermoscientific compound discoverer. What we appreciate with this software is that we can create our own workflows and specialize the way we want data to be processed. In the concrete case for us, this includes the use of MZ Cloud to support identification of unknown compounds. There is a very nice MSN library of spectra in there. We also are using MZ Vote for creating our own library. And finally, we also scoring everything with MZ Logic. We have a large volume of data, so a software that it's user-friendly and easy to use that does most of the work for us. It's amazing. We have our own workflows that are set up, and we just take the data, run it through the same workflow or the usual workflow, and we get our results.
With the range of new and exciting analytical techniques that we have available to us at the Centre for Metabolomics Research, we are really confident and really excited to be able to answer many of the biological questions that are of interest to our research groups in the very near future.